Hello, everyone, and welcome to today's webinar, panel presentation, Multiplex RT-PCR to accurately detect and differentiate respiratory viruses presenting similar clinical symptoms. This webinar is a part of 2021 Clinical Diagnostics and Research. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to thermofisher.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the right of your screen. You can also participate by submitting any questions you have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window below the presentation window and follow the process to obtain your credits. I'll now present today's speakers. Lauren Miller, Scientist, Clinical Applications, Marketing Support, Thermo Fisher Scientific, and Anami Patel, Ph.D., Chief Science Officer, Poplar Healthcare. For a complete biography on our speakers, please visit the Biography tab at the left of your screen. Lauren, you may now begin your presentation.
This winter, the United States healthcare system faces a special challenge. A potentially severe influenza season is expected to coincide with continued circulation of SARS-CoV-2. In this scenario, it's critical to have a single multi-target assay which can detect and differentiate between these viruses. The Takpath COVID-19 Flu A Flu B combo kit is an ideal solution for researchers and healthcare providers. So I'd like to start with two key concepts. Number one, the similarities and differences between SARS-CoV-2 and influenza. And then secondly, I'd like to talk about the implications of co-circulation of these two viruses this flu season. So number one, when we're comparing and contrasting the two viruses, the COVID-19 pandemic is an infectious disease caused by a novel coronavirus that emerged in late 2019. And the graph that you're seeing here is data from the World Health Organization of COVID-19 cases around the world from January 1st to today. And you can see the different regions are depicted with different colors. So you can see that COVID-19 infection increases and decreases in particular locations due to factors such as emerging variants, vaccine distribution, and infection prevention policies, so shutdowns, quarantine, business closures. And until herd immunity is achieved, we really don't know what the epidemiological pattern of COVID-19 spread will be, whether it'll follow the seasonal pattern seen in other respiratory viruses or not. The seasonal flu, on the other hand, has been around for a while, so we know that the seasonal flu circulates in these cyclical peaks which coincide with the seasons. The flu season in the Northern Hemisphere is from October to May, peaking from December to February. And depending on the year, anywhere between 3 and 11% of the US population is thought to suffer from a symptomatic flu infection every year. So when we compare these illnesses, hospitalizations, and deaths and numbers between the two viruses, it's really important to remember that the COVID-19 situation is so new and it's changing so much that experts are still trying to estimate the mortality rate of COVID-19. So the current thinking is that it's thought to be substantially higher, possibly 10 times or more, than most strains of the flu. So when we talk about influenza, it's important to realize that we're talking about 4 strains or 4 types of influenza. There's A, B, C, and D. Influenza type C generally only causes mild illness, while influenza type D viruses are not known to infect humans. So really we focus on influenza A and influenza B. It are these viruses that cause seasonal epidemics of disease every winter in the US. Influenza A is further divided into subtypes, while influenza type B, we don't talk about subtypes so much as distinct lineages. For example, Victoria and Yamagata. What is the significance between differentiating between flu A and flu B? After all, the patient treatment is very similar, whether you have flu A and flu B. Both A and B have potential for mild to severe disease. We differentiate between the two because influenza poses an ongoing risk to global human public health, and testing helps us monitor the subtypes and strains that circulate each year. This is very powerful and very important for informing vaccine design, assessing antigenic drift and shift, and assessing the impact on different population groups. So taking a look at the viruses themselves, both SARS-CoV-2 as well as influenza are enveloped single-strand RNA viruses, distinguished by the glycoprotein spike embedded in the envelope. So here on the left, we can see the SARS-CoV-2 with its infamous spike protein depicted here in purple, and influenza here on the right. And instead of one spike protein, there are two glycoproteins that are kind of the definitive characteristics. There's neuraminidase and hemagglutinin. It's these two proteins on the surface of influenza virus that contain the major antigens targeted by antibodies. With both viruses being a single-strand RNA virus, both of these viruses mutate. And when we talk about SARS-CoV-2, we talk mostly about novel viral variants. So most of these mutations affect the S gene, and emerging variants are classified based on the risk for public health. So we're all familiar with the alpha, beta, delta, and gamma strains. So these variants have garnered widespread attention because the rapid emergence in populations and evidence of transmission and/or clinical applications. When we talk about influenza viruses, the mutation rate and the mutation mechanisms are similar, but we talk more in terms of antigenic drift and shift, again, because this virus has been around longer and we're able to track the strains much better. Influenza's high mutation rate means that acquired immunity no longer offers enduring protection against the whole class of influenza viruses. So new subtypes can be more virulent and potentially lead to higher mortality rates than their predecessors. Both the seasonal flu and COVID-19 are transmitted easily through contact droplets and aerosol, whereas COVID-19 is more contagious and there are more superspreader events. They have similar symptom onset and incubation periods. Although COVID-19 is really characterized by this longer incubation time, symptom onset can be anywhere between 2 and 14 days after infection. Similarly, for the incubation time when you're contagious, the seasonal flu is slightly shorter than COVID-19. And in both cases, patients can be asymptomatic or have mild, moderate, or severe diseases in both cases. So in terms of symptoms, COVID-19 and influenza are extremely similar. It's very difficult for healthcare providers to differentiate between these two viruses based on symptoms alone. Both of them can produce fever, chills, cough, sore throat, runny nose, headache, fatigue. And in both cases, they can lead to severe disease, such as pneumonia, acute respiratory distress syndrome, myocarditis, multiple organ failure, etc. And it's important to know that even though these two diseases have very strikingly similar symptoms most of the time, it is extremely important that healthcare providers diagnose influenza or SARS-CoV-2 infection by confirmation testing. For an example, there are some severe or certain symptoms or treatments that are very different between the two diseases. So a cytokine storm is one potential symptom of a COVID-19 case. And this is where excessive production of pro-inflammatory cytokines can lead to acute respiratory distress syndrome aggravation, causing widespread tissue damage, multi-organ failure, and even death. So the drugs used to treat cytokine storms are immunosuppressants, which of course are contraindicated for seasonal flu because it reduces the patient's ability to fight the virus. Therefore, despite the similarities of symptoms, when it comes to certain severe symptoms or complications, diagnosis and confirmation testing is critical. So now that we've talked quite a bit about the similarities and differences between these two viruses, I'd like to discuss the implications of co-circulation of these two viruses this year. So patients with acute respiratory illness symptoms can either kind of go one of two ways. They can require hospitalization, such as if you're old, high risk, or a nursing home resident, or you can have mild symptoms not requiring hospitalization. If you do require hospitalization, the CDC recommends that the healthcare provider orders a multiplex nucleic acid detection assay for influenza A, B, and SARS-CoV-2. So in this situation, we can see that the Takpath COVID-19 Flu A, Flu B combo kit is an ideal solution for researchers and healthcare providers. And even for those not requiring hospitalization, the CDC still recommends testing for SARS-CoV-2, as well as testing for influenza if the results affect clinical management or for infection control. It's important to note here that SARS-CoV-2 and influenza virus co-infection can occur. In looking at the slide overall, it's easy to imagine the increased burden that this could have on the United States healthcare system. With a higher number of severe influenza illnesses, the hospitalizations and intensive care unit admissions would all compete with 16 COVID-19 cases. So if you think of, doctor or nurse burnout, it's the same doctors and nurses who would treat either patient. And even for those not requiring hospitalization, we would see an increased demand for testing. It used to be that if you had mild symptoms for a seasonal flu in the winter, you would just go home and get the bed rest. But now we need to get tested for COVID-19. It's extremely difficult to tell the difference of, is this a cold, a flu, or the onset of COVID-19? Looking at the treatment, so with co-infection of these viruses, or co-circulation of these viruses, it's important to understand that even though the symptoms as well as mild management may be similar, treatment options for severe or high-risk cases are very different. So in addition to the cytokine immunosuppressant example that we saw before, overall we can see that treatment for severe cases are extremely different, whether it's COVID-19 or flu. So COVID-19, there are monoclonal antibodies that can be effective as long as they're initiated early on in the course of infection. And for the flu, we have antivirals such as Tamiflu, which can be extremely effective when given within the first 2 days following influenza symptom presentation. And what I want to point out here is that even though these treatments are independent of viral variant or subtype, of course, they're very different between each other. So you can't use influenza antivirals to combat a COVID-19 infection and vice versa. With these two co-circulating viruses, it's also important to mention the importance of vaccination. So both of these viruses are pretty effectively inhibited by public health safety measures, hand washing, covering your mouth, covering your sneezes, wiping down surfaces, avoiding contact, social distancing. But it's really the vaccination that provides the protective, important protection from COVID-19 and influenza. COVID-19 vaccines are now available. There are a few different authorized for use under EUA. It's recommended for everyone at least 12 years or older. And the CDC is now recommending that certain people be eligible for COVID-19 booster shots. The flu vaccine, on the other hand, is updated every year based on the surveillance data of influenza viruses detected in the previous season. There are several vaccines that are FDA licensed, and they're recommended particularly for high-risk groups. The thing that I want to focus on is really the updating annually based on last flu's data. And the reason that that's important is because we virtually had no influenza season last year. So if we take a look at this data, this is the circulation of flu A and B in the United States. And we can see this cyclical pattern every year, rising and falling, where we would expect to see influenza season, until last year, where it virtually drops off into nothing. So this red rectangle here on the right indicates the beginning of the COVID-19 pandemic. This is also the beginning of social distancing, stay-at-home orders, school, venue, and business closures, bans on public gatherings, travel restrictions, contact tracing, and quarantine. So we can really see that the effectiveness of all of these measures against COVID-19 is also extremely effective against other respiratory illnesses, such as influenza. And even though it looks like there's just a flat line for the influenza last year, if we zoom in, we can see that there was, in fact, a flu season. It was just very small, on a much, much smaller scale than normal. So I want you to take two things away from this. Number one, We have extremely limited amount of data from last year's influenza strains to inform this year's vaccine designs. And number two, this record low activity means a lower population immunity, which puts us all at risk for increased disease this year. So this is a graph kind of showing the easing of public measures during the COVID-19 pandemic. So this graph on the left, December 10th of last year, before the vaccine became available, in this dark red coloring, you can see that most countries around the world had very stringent protective measures. Since then, you can see these lightening of colors, so kind of this global easing of protection measures, slowly but surely as the world starts to open up. So this easing of social distancing measures is what experts believe might lead to a surge of influenza infections. So just to recap, our concerns for the 2021-2020 flu season are these. Higher activity of influenza virus is anticipated. Easing social distancing and other measures may lead to a surge in infections. Decreased population immunity due to non-pharmaceutical intervention and lower exposure to influenza in the previous season. And limited data available for influenza strains in the previous season for vaccine design, which increases the likelihood of influenza vaccine mismatch. So overall, testing recommendations from expert groups around the world, such as the European Center for Disease Prevention and Control, as well as the Academy of Medical Sciences, is basically in strong support of multiplex testing for both SARS-CoV-2 and influenza during this flu season. Running multiple tests on each patient is difficult and it's expensive. Combining targets for these viruses into a single test makes distinguishing between these infections much easier and can help physicians choose the appropriate treatments and protocols for their patients. And this being said, the Takpath COVID-19 Flu A Flu B combo kit is an ideal solution for healthcare providers and researchers. And with that, I'd like to introduce Dr. Anami Patel for clinical performance of the Takpath COVID-19 Flu A Flu B combo kit as compared to other multiplex RT-PCR tests.
Thank you, Lauren, so much for a nice introduction. I work here at Popular Healthcare, which is part of Path AI, located in Memphis, Tennessee. Popular Healthcare is a centralized laboratory for histology and core diagnostic work, providing services to all 50 states within U.S. We do offer comprehensive molecular diagnostic testing along with other pathology services to our clients, including primary care physicians and private practices throughout the country. Here, as we know, when we were hit by pandemic in spring of 2020, Popular Healthcare and our group immediately started looking into offering this COVID testing, and we were very fortunate to develop our own assay, CDC-based, and apply for FDA emergency authorization use. So once we validated and applied for EUA, that was around early April 2020, and then immediately we started building our capacity by looking into different platforms, PCR, and reagents and supply, as every other lab went through that. So currently we offer SARS-CoV-2 diagnostic test by using different platforms and reagents such as Thermo Fisher, Takpath, COVID assay, Roche Cobas assay, and Applied Biocode, and another one, Hologic Panther assay. So that way we have good capacity and good service around SARS-CoV-2. In addition to performing laboratory testing, we also have here at Popular Healthcare our own collection site drive-through where we can help our community, where people would come, register, and we collect the test and offer the testing at the same place. And lastly, we have been doing surveillance testing since beginning of this year by whole genome sequencing, Illumina sequencing platform. So this is very comprehensive SARS-CoV-2 capacity and testing we have developed over the last 18 months or so. So where we are now, what we are up to, we are obviously getting ready for upcoming flu season. As Lauren mentioned, last year we did not see any influenza activity, very evident from her graph, and the several reasons behind as well. So several months ago, here in our lab, we started looking and preparing for upcoming influenza season. And why we were interested? What was the reasoning behind that? Since we have been doing comprehensive respiratory pathogen panel testing for several years now, and during pandemic, upcoming influenza season, there was a need. Our clients, hospital clients, urgent cares, nursing homes, they started asking us, and the reason be also the symptoms, which Lauren explained very well, that it's hard to differentiate somebody who has a respiratory infection or symptoms, whether it is SARS-CoV-2 or influenza A or B. So there was a need to look into, and at the same time, CDC last year, around August, September month, they put in their clinical guideline the strong recommendation around testing for influenza as well as SARS-CoV-2. And they were able to get FDA emergency authorization use for their first test. And as we know, in 2020, we didn't see any influenza, and because of the good measures and other things which was discussed earlier. So given this uncertainty, our lab decided to look into multiplex test where we can use the same, keep the same capacity and perform the test and give the results as a combo, either influenza or COVID, rather than two different tests. Is there some multiplex testing we could do? So as part of that, the single test can use up the capacity, as I just said. So the combo test can allow lab like us to process more tests in a given time period using the same amount of reagent. And then at the same time, it can help control the spread of not just SARS-CoV-2, but influenza virus as well, and helps ongoing surveillance support for SARS-CoV as well. And importantly, very importantly for us, the testing material, what we are using, and sometimes here and there, we still see some shortage of the supply, especially around consumables. So we looked into what we have and combo test how we can adapt. So we looked into our within our lab what capacity platforms wise and training, expertise, staffing, and so three things came to our mind. Three platforms: Thermo Fisher, Roche 6800, which we have in our lab, and Applied Biocode MDX 3000, three different systems. And these three platforms are already available combo test as FDA EUA. So first, just showing you the workflow around Thermo Fisher Takpath COVID-19 influenza A and influenza B combo test performed on 7500 fast DX. So once we receive the sample in VTM or saline, then we prep the sample. What that means is we extract the nucleic acid as part of the workflow. And once the nucleic acid is extracted on Kingfisher instrument, which can do up to 94 patient samples on a given run, plus two external controls. So once 96 samples are being extracted, it can go on 7500 fast DX instrument, where we can run either 384 samples or 94 samples plus two controls. And there it requires the extraction part, the prep sample part requires around 30 minutes. And then once we set up a PCR and put it on that 7500DX instrument, which would take around hour and 4 minutes. And then it has a software built in onto the instrument where we can analyze, review the result. And this entire workflow in our lab for SARS-CoV-2 as well as combo test, it is bidirectionally interfaced. So order can come through our LIMS system into the instrument. We can process a sample, review the result, and release the result in our own LIS system. So, and the other two workflows, which I mentioned, the instruments we already have in our lab, Cobas 6800, we evaluated, as part of this study, we ran the samples on 6800 system with Cobas reagents for influenza A, B, and SARS-CoV-2. And in that workflow, it requires a sample would come into the lab. We would transfer 600 microliter of the sample into another tube in the hood, and that will go on 6800 analyzer. That's where, and along with the sample we would load reagents for this assay as well as the consumables, required consumables. And it will process the sample, so the extraction, PCR, amplification, and detection would happen on that run. Again, we can run 94 samples and 2 controls on a given batch, and then next set of samples can go on after 30 minutes or so on 6800 system. On the third system, applied biocode, where we would need to extract the samples on a Kingfisher instrument, which is from Thermo Fisher. And then once samples are being extracted, in this case RNA, then we would set up a PCR and it will go on applied biocode MDX 3000 system. And amplification and detection, again, FDA, EUA assay. So both of this workflow, again, requires around 3.5 hours. So time-wise, it is about the same process. On this slide, I just put all 3 platforms side by side. And for clinical evaluation of Takpath COVID flu A and flu B test, which we were very interested in, and compare with Cobas 6800 and applied biocode in our lab, we were able to retrieve previously tested positive and negative sample. So here in this study, we were able to get 60 SARS-CoV-2 SARS-CoV-2 samples positive, and these samples were from September of 2021. And I emphasize that because this positive samples, we were able to, as part of our ongoing surveillance process, we had access to the data result on the variants, and most of these samples which were included in this study were Delta variants. So, and then we were able to get 60 influenza A positive samples from 2019 influenza season, and that time as a clinical standard of care test, we were running on BioFire instrument. So those 60 influenza positive result from BioFire RPP panel, we were able to run on combo test by all 3 methods. And then limited number of influenza B, 46 positive samples. And then another set of 60 negative samples for all 3 pathogens we included in this study. So let's look at the data, how it performed. So before that, I'm just trying to show you here, whenever we do this kind of clinical evaluation with different reagent and workflow, we want to be inclusive with our samples, not just certain number of positive, but how diverse they are in terms of viral load. And in this situation, viral load would be based on the CT value. So, and this CT value is once we ran the sample, what CT value we had between each platform for combo test. So here in this case, on the upper left top graph where it shows us influenza A CT value, and dark color is thermo combo test, and light blue color is Roche Cobas. So they're pretty good representation between different CT value or viral load or severity of infection. Similar way for influenza B, those 46 cases, and for COVID-19, another set of 60 samples. We were not able to put this information for applied biocode, and the reason being that platform gives us MFI value rather than CT value, which doesn't correlate with either viral load or severity of infection. So I'm just showing here the comparison between Thermo Fisher and Roche Cobas system. So let's look at how it performed. And first 60 set of data, influenza A, and we evaluated in terms of positive predictive value and negative predictive value. So positive predictive value, it gives us an idea around how sensitive clinically. So if patient had infection and we tested with any diagnostic assay, was it able to test positive or not? And that would depend on the limit of detection or analytical sensitivity. We have not done any analytical validation or analytical performance between these three reagents because it is very well documented in each of their package insert. We were mostly interested in how it performs clinically with clinical samples in our lab. So for influenza A, when we ran and then upper left graph shows comparison between Takpath COVID influenza A and B combo kit versus Cobas combo kit. So 60 positive samples gave us results by both method positive, and altogether 170 samples we ran for, which were negative by influenza A. So all that matched. Now remember, we only ran 60 negative samples for all three targets, but here 170 numbers include influenza B negative, influenza B positive result would consider as influenza A negative. So that way number is higher for negative predictive value. So in this case, the positive and negative predictive value compared between Takpath COVID combo test and Cobas SARS-CoV combo test is 100%. Similar way between applied biocode and Cobas, other two methods, similar result 100%. And when we compare Takpath versus applied biocode, again, positive predictive value and negative predictive value were 100%. When we ran total 46 positive samples for influenza B on Takpath COVID combo assay, we were able to get all 46 influenza B for positive, as well as on Cobas combo test. Similarly, when we ran on applied biocode with applied biocode reagents, all 46 were positive on their platform, and Cobas 100%. And comparison between Takpath COVID and applied biocode was 100% as well for positive and negative. That gives us the clinical sensitivity and specificity at 100%. So Now, when we looked at the results for 60 positive, in this here we had 61 positive for SARS-CoV-2, and all 61 match between Takpath COVID and Cobas, as well as Applied Biocode and Cobas, and Takpath COVID and Applied Biocode COVID. So remember, these are different data I'm presenting, but by running one sample, since it's being all three assays being combo test, we get all three results same time. Except on Applied Biocode, their test method is first you run influenza A and B, and from the extracted nucleic acid RNA, you set up another assay for COVID. So it would require two separate runs. Other than that, everything is similar. You run for Roche Cobas and Thermo Fisher Takpath only one time, and you get all three results. So one thing we noticed that on Applied Biocode, one of the COVID positive results failed or invalid because of the internal control. So in that situation, we did not have one result, valid result. So in conclusion, it was not much very exciting data. In a way, it is a good thing for lab. So the study was completed with 100% agreement between all three assays. And we saw that they all equally perform well and effectively for our infectious disease testing. And multiplex PCR test can save both to the lab and to the patient significant headache. In this case, we see a lot of people coming to our drive-through have respiratory symptoms, and they are just concerned about or stressed out around COVID. And when we test, definitely COVID is in some cases is not positive. But at that time, the order was only for COVID, and we ran only for COVID, so we would not know whether any influenza was detected or not. So, and lastly, I would say that with more viruses entering into our ecosystems, multiplex testing is very trustworthy route to differentiate between different infections for effectively managing the patient. And here I'd like to first thank you, Thermo Fisher, for giving us opportunity to evaluate and Popular Healthcare R&D team here, specific to Lee Wink, one of our R&D scientists, To Gang, our supervisor for molecular infectious disease, and Mia Trong, who helped carrying out this study. And happy to hand back to Lauren.
Great. Thank you so much, Dr. Patel, for sharing your data and your experiences. I'd like to spend the last 10 minutes just wrapping up with an introduction to the Applied Biosystems Takpath COVID-19 Flu A, Flu B combo kit. So this Takpath COVID-19 Flu A, Flu B combo kit is an all-in-one real-time PCR respiratory test. So this is a single test for SARS-CoV-2, Influenza A, and Influenza B. It simultaneously differentiates between the respiratory viruses and helps identify cases of co-infection. This is an affordable and scalable solution, increasing testing throughput and lab efficiency. It has sensitive and specific RT-PCR detection, boasting a robust performance for detection of COVID-19 flu A and B. And the results are automated by the pathogen interpretation software, which quickly interprets results to expedite reporting and reduces risk of user interpretation error. This kit is available in the US under FDA UEA emergency use authorization only. And for more information about the product details, you could see more at our product landing page. But just to mention, The kit targets the S and N genes of SARS-CoV-2, the matrix genes of influenza A, and the matrix genes of influenza B. We do have an MS2 internal control, as well as a positive control. And we'll go over the authorized sample types, sample prep, kits, and systems in the next few slides. So this is the authorized workflow that Dr. Patel briefly had an overview of, anywhere from receiving samples to sample prep to real-time PCR to data analysis. The Takpath COVID-19 Flu A Flu B combo kit is a simple, modular, and mostly automated workflow that user can easily add to their existing COVID-19 testing menu and existing infrastructure to expand their respiratory sample testing while maintaining a low operational cost and workflow simplicity. So we'll go through each of these stages of the workflow in a little more detail, starting with receiving samples. A clinical laboratory receives samples collected from nasopharyngeal swabs or interior nasal swabs. Next is an automated sample preparation step. So in this step, the MagMax viral pathogen 2 nucleic acid isolation kit is used on the Kingfisher Flex purification system with the 96 deep well head and deep well heat block. Next, there's a manual qPCR step, followed by an automated qPCR step using the Takpath kit on either the 7500 FastDX real-time PCR system or the QuantStudio 5 real-time PCR system. Finally, data is analyzed using the Applied Biosystems pathogen interpretive software. So not only does the software automatically interpret test results, but it also automatically performs QC for all controls on the plate and alerts users for the completion of data analysis. It generates interpretive report, including each specimen, and generates lab configurable exports for laboratory information system integration, such as the one that we heard for in Dr. Patel's presentation. So all in all, in the context of co-circulating SARS-CoV-2 and influenza, running multiple tests on each patient can be difficult, time-consuming, and expensive. Combining targets for each virus into a single test makes distinguishing these infections much easier and can help physicians choose the appropriate treatment and protocols for their patients. The Takpath COVID-19 Flu A Flu B combo kit is an ideal solution for researchers and healthcare providers, with its end-to-end workflow taking about 3 hours for 94 samples. This is an effective solution, and you can learn more at thermofisher.com/COVID-19 flu. Okay, so now to get into performance characteristics just a little bit. So the limit of detection study was established to figure out the lowest viral concentrations that can be detected by the kit in at least 95% of cases. Negative nasopharyngeal swabs were pooled and spiked with the indicated strains of virus at a range of concentrations and processed through the workflow. And what we saw is that for SARS-CoV-2, a minimum of 100 genomic copy equivalents per milliliter were detected. And for influenza A, between 200 and 500 GCE per mil, depending on the strain, were detected. And for influenza B, again, depending on the lineage, between 500 and 1,000 GCE per mil were detected. Now, when we look at this limit of detection compared to our competitors, we can see our limit of detection here in red for influenza A, B, as well as SARS-CoV-2, compared to those of some competitors. So we have the Cobas and Cepheid Gene Expert here in green. We can see that for influenza A, our limit of detection outperforms competitors, while for influenza B and SARS-CoV-2, our limit of detection is in the range with the competition. So our inclusivity data. In vitro analysis was performed for influenza A. Functional testing was performed to determine the lowest concentration that would produce positive results in all replicates. And then it was found that in 100% of cases, all 10 of these common strains were detected for this lowest concentration. Similarly, for influenza B, we can see that all 5 of these common influenza B lineages were detected 100% of the time at these low concentrations. Here's the competitive interference study. So for this study, negative nasopharyngeal swabs were pooled, and then they were spiked with combinations of SARS-CoV-2, influenza A, and influenza B. In certain combinations where one virus was at a very low concentration, one was at a very high concentration, and one was absent. So in these cases, as you can see, no competitive interference was detected. And you can also see very high sensitivity as well as high accuracy for the virus that was spiked in at a low concentration. And so from this study, we can tell that this test is very accurate, even in the presence of co-infections. And finally, the clinical study results. So in a clinical evaluation study was carried out on archived nasopharyngeal specimens. These samples were tested using the Takpath COVID-19 flu A, flu B combo kit, as well as 2 FDA-approved comparator tests, both for SARS-CoV-2 and influenza. And you can see here that the positive % agreement and negative % agreement exceed the FDA agreement for performance guidelines. Okay, and then the very last thing that I want to mention is that there are multiple multiplex testing solutions for COVID-19 and the flu. On the left, we see the kit that we've been talking about today is Takpath COVID-19 Flu A, Flu B Combo Kit. This is a single test that detects and differentiates between the two viruses with a high sensitivity and specificity. And then on the right, we can see a Takpath COVID-19 Flu A, Flu B RSV Combo Kit. So this is essentially the same test, but with RSV included in this test, which helps diagnose and monitor the spread of this respiratory virus as well. This also boasts a high sensitivity and specificity with robust performance for detecting SARS-CoV-2, influenza AB, as well as RSV AB. These products are highly regulated and not approved in all countries. Please visit thermofisher.com/COVID-19 IFU for all country-specific instructions for use. And with that, that's all I have. Thank you very much.
Thank you, Lauren and Anami, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Let's get started. Our first question is, how accurate is a multiplex test versus a one-virus test?
I'm sorry, who is this question for? Is that for
It's not. It doesn't say either way.
Okay, well, I can definitely chip in first. So I would say, in terms of multiplex PCR, there are certainly some cons when using a multiplex PCR for very, very large panels. There can be competition for reagents. There can be primer interaction for very, very large, very complex panels that you can certainly see a loss in sensitivity due to that. But for panels such as this, it is a multiplex, but there's so few targets that we really don't see that drop in sensitivity. And then also, for commercial assays, those things are tested very robustly. So things like competition for the master mix or things like that, that's very thoroughly investigated. So for our flu A, flu B, COVID-19 combo kit, you can see in our clinical evaluation, for example, we tested our kit with single virus tests, and you saw the above 95% sensitivity. So it still remains very accurate. Dr. Patel, I saw you put your hands up if you wanted to add some information or add anything to that.
Yeah, sure. So we, after this study, the data presented here, we look at the same samples by just SARS-CoV-2 assay we ran, and we did not see any significant difference. Positive remained positive, so we did not lose. And at the same time, the crossing threshold between combo and single test were very close. No statistical difference.
Great, so thank you. Next question, are co-infections a real possibility?
Go for it.
Yeah, as far as co-infection, yeah, that's a very good question. I think, yes, it is possible to have flu as well as other respiratory illness and COVID-19 at the same time. Um, as we are where we are right now, health experts are looking and studying how common this can be. We don't know. We don't have a lot of data yet. But some of the symptoms of the influenza and COVID are so similar, and it does make it very hard to distinguish based on symptoms only whether it is influenza infection or SARS-CoV-2. So the test would be in that situation helpful to differentiate between influenza and COVID.
Yeah, I absolutely agree. And just to add the fact that, there is very, very little epidemiological evidence of COVID or flu and COVID co-infection, just because there's such little testing of the flu, since there was hardly any flu season last year, and because the COVID-19 pandemic, not many people are actually testing for this, but you see some studies done on this co-infection, and it's a pretty wide range of results worldwide, because it depends so much on geographic location as well as the prevalence of influenza at that time and at that place. So, you'll see some people do studies during an influenza outbreak, and of course those numbers will be very different than someone doing a study just on retrospective samples, kind of more out of curiosity. So I've seen anywhere between, in certain population subgroups, 0.2% of all positive COVID tests will also test positive for influenza, whereas in other places at other times, like, for example, in Wuhan, China, during the first outbreak of COVID, I think it was up to 54% co-infection with influenza. And then in parts of California right now, I know that the California Department of Health published a paper where about 23% of positive COVID cases also tested positive for influenza A. So, this range of 0.2 to 50% really kind of demonstrates the fact that it is very possible, but it is so highly dependent on where you're testing and who you're testing.
Thank you. Our next question is the interpretive software included in the kit for either of you?
So for the Applied Biosystems interpretive software, when you purchase the kit, you purchase the kit from the landing page for the combo of the COVID Flu A, Flu B kit. And then there is also a customer support contact number that you contact for your software. So you would just go to the thermofisher.com website, or you can also go to thermofisher.com/contact us, and that will give you a direct link to customer support and the service center, IT support, and that's where you get in touch with a representative, and they will hook you up with the interpretive software.
Great, thank you. Next question, how can I order the kit?
Yep, same thing. So you can go to the thermofisher.com and enter I believe the catalog number is A49868. So just search for the Takpath COVID-19 Flu A, Flu B combo kit. And from that page, you'll be able to request a quote. Alternatively, you can go to that same page, the thermofisher.com/contact us page, and work with your sales representative to order a kit.
Great, thank you. We're going to wrap up with this last question. This is for Anami. How much hands-on work was involved in running the 3 assays?
Yeah, so for running Thermo Fisher Takpath flu and COVID combo assay, again, if I break down by processing the sample, extracting RNA on Kingfisher, so hands-on time for full plate, 94 samples and 2 controls, about 30 minutes. And then on Kingfisher, it takes about 20 minutes. And then another 15 minutes or so to set up PCR plate, and then it goes on studio. So here, and then after PCR is done, analytical software will generate the data excuse me. And it takes about another 10 minutes to review and release, so less than 1 hour. And similar thing on applied biocode, but on Roche Cobas, I would say maybe 30 to 40 minutes hands-on time.
All right. Well, thank you, Lauren and Anami. Do you have any final comments for our audience? Oh. All right. Well, thank you again, Lauren and Anami, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsors, Thermo Fisher Scientific, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until November 2023. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.